Here we are in Chapter 5, Energy, Enzymes, and then specifically in this video we'll be going over how membranes, their role in the chemical reactions that occur inside cells. We're going to focus on membranes and the role that they play in providing structural organization for chemical reactions or metabolism. So they're the barrier that separates the inside and the outside environment of a cell. They provide a location for chemical reactions, and they harbor several different macromolecules that we talked about in Chapter 3. So if we were to look very closely at the plasma membrane of a cell, we're going to see that in this picture, you can see that this clear little barrier right here is in fact the very thin membrane that distinguishes between the inside and the outside of a cell. The cytoplasm itself is very darkly stained in this picture, and that is because the cytoplasm is full of substances and it is very, very complex. It's not just a clear liquid as it's previously been described. So what the membrane does is it decides what can get in or out of a cell. The cell membrane is composed up of phospholipids. Again, we talked about these in Chapter 3. Phospholipids form two layers or a bilayer. And phospholipids are really cool because they have a complex structure that has a hydrophobic or hydrophilic head. And hydrophilic head means that it loves water. So the hydrophilic head here with its phosphate group is going to be in an aqueous or water-based environment, whereas the hydrophobic tails, whether they're saturated or unsaturated, are going to be hydrophobic. Phobic means you're afraid of the water, and so they're going to be tucked inside. And because of this combination of hydrophilic and hydrophobic, what we find is that the membrane itself forms two-layer or bilayer sheet. So here we have all the hydrophilic or water-loving heads. They're facing out or in to the watery environments that are found outside and inside of a cell, whereas those hydrophobic tails are tucked nicely inside those hydrophilic heads where they're not in contact with water. And so the entire structure is happy and well put together. There are two different types of hydrocarbon tails that are found on the phospholipids. This should be a review from Chapter 3, but what they do is they determine how fluid the plasma membrane is. And so if you see here where we have lots of unsaturated hydrocarbons, so those unsaturated hydrocarbons kink because of the double bond between two carbons. Each one of these kinks represents an unsaturated double bond. And because of that, it pushes the two phospholipids further apart, creating a very fluid plasma membrane. Whereas if all of the phospholipids are tightly packed together, all the hydrocarbon tails are saturated and they're linear, then everything's very tightly compact and you get a very viscous or solid cell membrane. Now, previously we have taught you that the phospholipid bilayer contained proteins, and that was the sum total of the cell membrane. Here we need to realize that it's more complex than that, and that there are actually steroids, which are also a lipid, that are embedded within the hydrophobic region of the phospholipid bilayer, and their job is to actually lock the phospholipids in place. So each one of these ring-like structures represents a steroid cholesterol, and that cholesterol holds these two phospholipids together with each other so that there's less likely to move, which then makes the membrane less fluid. So it turns out that the steroid cholesterol actually has an effect on the fluidity of the membrane, and it actually changes depending upon the temperature at which the cell membrane is at. Then here's the more complete picture of the cell membrane. So if you look closely right here, you'll see these darker shaded areas in the phospholipid bilayer. Those are actually the steroid cholesterols embedded. You'll see these ginormous purple structures. Those are the proteins that are found in the phospholipid bilayer, and this is why we call it a mosaic model. And the proteins you'll see will interact with carbohydrates either on the outside or the inside of a cell. And so there are glycoproteins where the carbohydrates are attached to one of the proteins in the membrane, and there are glycolipids where the carbohydrate is directly bonded to a phospholipid within the membrane. Of course, all of this interacts with the extracellular matrix or the ECM on the outside of the cell. The proteins that are found in the phospholipid membrane, there's several different types. The first type is the integral proteins. And what integral proteins do is they actually are embedded entirely within the phospholipid bilayer. So this protein is not only visible or kind of structurally hanging out on the outside of the cell, it hangs out on the inside of the cell, yet the entire structure still penetrates the double layer. Now this is important because in order for the protein to do this, it has to contain amino acids right here in this structure that are hydrophobic, just like these hydrocarbon tails of the phospholipid. Whereas this region, sticking up on the outside and the inside of the cell, have to contain amino acids that are hydrophilic or water-loving because they're in contact with the outside and inside aqueous environment. And so this protein structure is only possible based on having the correct sequence of amino acids within it. And you can see this one has the alpha helical structure, or it could be a beta pleated sheet. But again, this one's an alpha helix because it's twisted. There are proteins that do a lot of functions. Some of them are enzymes, and enzymes we just talked about in our previous video. Enzymes do chemical reactions by providing a binding site. If you see in this video, we're showing that the reactant or substrate comes in, it turns through a chemical reaction into a different product. That product then goes in and becomes a substrate of a neighboring protein, which then creates its own unique product that can go do its own chemical reaction within the cell. Then there are proteins that actually allow for signaling, and we'll talk a lot about signaling when we actually take a chapter from the other red AP Bio book. But what this does is you can see that this integral protein, it actually has a receptor for a molecule on the outside of the cell. The molecule never actually enters the cell. The signal, though, is simply transferred to an integral protein within the inside of the cell, which then activates a molecule on the inside of the cell. So a message from the outside activates a signal on the inside, and that molecule can now go do its own function. There are membrane proteins that will not only be enzymes, but they'll do transportation. And so here we're going to move substances across the membrane. So we simply provide a channel for which molecules can move through. Sometimes it doesn't require energy, and sometimes it does. Some of the transportation of molecules across the membrane does not require energy. It's called passive transport. Passive transport, we're going from an area of high concentration to an area of low concentration, from very crowded to less crowded. So if we watch the progression over time, eventually we'll reach equilibrium or equal amounts on both sides. The same thing happens for every molecule in its own right. So here they're showing that this was high concentration of green, and here's the high of purple. So the purple is going to move in the opposite direction of the green simply because it has its own concentration gradient. Again, though, they just want to reach equilibrium. There are some molecules that are able to diffuse or do passive transport directly through the phospholipid bilayers. Most others have to go through a protein. Oxygen and carbon dioxide are two of the best at going through the phospholipid bilayer. Other small, nonpolar, or hydrophobic molecules can easily do that. A lot of times we're talking about lipids. Very, very small lipids are able to easily diffuse through the phospholipid bilayer because like dissolves like. And lipids are what make up the bilayer, so lipids are able to get through the bilayer. There are transport proteins that will help in the diffusion from high to low. So if a molecule is not able to get through the phospholipid bilayers, it's going to wait and go through a transport protein. And so these provide passage. Because the protein is helping them move through the membrane, we call it facilitated diffusion, because to facilitate means to help. The transport proteins are actually made in the ER in the Golgi apparatus within the own cell. So here's our endoplasmic reticulum. It's producing the protein. The protein is transported to the Golgi body. The Golgi body modifies it. And then when it secretes the vesicle, you'll notice the vesicle has its own membrane around it. It's the exact same membrane that makes up the cell. And so it's going to fuse. Once it fuses, its membrane just integrates itself into the cell membrane. And so we have to realize that the cell membrane is constantly changing. It's getting bigger, it's getting smaller, and it's fusing or pinching off small parts. And so it's constantly being recycled and altered. It's not a very rigid structure. One of the specific examples of diffusion across the membrane that requires facilitated transport is osmosis, or the movement of water. Water will always go from an area of high concentration to low concentration. However, when we talk about it in science, we don't talk about it as water going from high to low concentration. We talk about it as water going from low solute concentration to high solute concentration. And the reason is that if you look, if we have lots of water and very little solute, then we have low solute concentration but high concentration of water. So water is going to go from where there's lots of water to where there's less water. And because of that, we have to make sure we keep it straight in our head that low solute means lots of water. Now, the osmosis, or movement of water, is really important because it's what regulates all of our cells in the environment. So if we look here, we can see what happens if you put a cell into a specific environment. So in an iso or equal solution, the cell water will go in and out at an equal rate. Nothing will really change. It maintains. However, if you put the cell into a hypotonic solution, which means lots of water outside, very little water inside, what happens is the water comes into a cell. In an animal cell, it's going to burst it, or what we call lyse. Whereas if it goes into a plant cell, the plant cell has that cell wall. And so the plant cell is just going to push and push and push against the cell wall, but it has nowhere to go, and so it becomes turgid, or very rigid. Turgidity is what allows plants to stand upright in their environment. If we were to put our cell in a hypertonic solution, which means there's lots of solute outside but very little water, the water is going to leave our cell. And when that happens, our cells will shrivel up. However, a plant cell, it will shrivel up, but the cell wall is rigid. It can't move. And so what happens is we call this to be plasmolysis, or plasmolyzed. And so you can actually see that the cell is pulling away from the cell wall. It is a structure that you can see in the microscope. We will look at it in lab. So the whole point of maintaining the amount of water is to have osmoregulation, to coordinate that. Now, not everything can happen without the input of energy. And so if we want to move things across the cell membrane, sometimes we need to do or use ATP for that. And so this is active transport. So some transport proteins are actually going to move things from low to high. They're going to force it to become more crowded, and that serves a critical role in communication. So when the solute goes from low to high, we're going against a concentration gradient, and it's only possible through the use of ATP. So what happens is we have our solute. It binds to the active sites with the help of ATP. ATP phosphorylates the enzyme protein, which then allows it to change shape. So now that the protein has become phosphorylated, it changes shape, a chemical reaction happens, and we're going to release our solute to the outside to the high concentration. And notice that the protein went back to its original shape once the phosphate detached itself or has become dephosphorylated. An example of active transport kind of on a bigger scale is exocytosis and endocytosis. Exocytosis is where we are exiting or having a vesicle fuse with the cell membrane to release large particles, whereas endocytosis is where we're taking them in. Again, notice that the vesicle cell membrane is just inserting itself into the cell membrane itself. Now, one thing about the infolding or the endocytosis is that there's specific types of endocytosis. There's phagocytosis, which is what we typically associate with amoebas. They have these cell membranes that look kind of like arms. They're not really, but they look like arms. They reach out and they grab and they engulf large quantities of food. That's phagocytosis. Whereas pinocytosis, you can see this infolding, these little bubbles right here. This infolding is where we are actually creating a pocket, taking some of our own cell membrane to encapsulate some liquid molecules and then bring them into the cell as a vesicle. The third type is receptor mediated. So it's the exact same process as the endocytosis or pinocytosis. However, we're requiring a receptor protein to be on the cell membrane to trigger this process. One of the best examples is cholesterol. So what happens here, this is an example of receptor-mediated endocytosis. Here's our cholesterol wrapped inside of a vesicle, and it has proteins on its structure. These proteins will specifically bind with the receptor on the proper cell membrane. So the protein comes in and binds. Once this receptor-mediated binding occurs, we're going to trigger the endocytosis of this entire vesicle. If you're missing these receptor proteins, your cholesterol will never enter cells, and therefore you'll have high levels of cholesterol in your blood, which then can cause kind of special health risks. And we're done.